We thank everyone for joining us today. We are pleased to welcome Professor Dr. Stefan Tenzer from the University Medical Center of Mainz. Stefan has been a longtime user of PEAKS and is well known in the proteomics community. His research career has begun at Eberhard Karls University of Tübingen, Institute of Cell Biology, and in the Department of Immunology, he obtained his PhD in biochemistry. After graduating in 2004, Stefan has begun working in Mainz and has had a very successful career and taking on various positions across different disciplines. From applying mass spectrometry-based techniques for antigen processing as the group leader at Johannes Gutenberg University of Mainz, to being appointed at the head of mass spec core facility of the Research Center for Immunotherapy in 2009. As a well-developed lab, the Tenzer group has applied mass spec methods in other research areas by joining the Research Center for Translational Neuroscience, as he is leading the PROTIC unit, which is proteomics and transcriptomics. Most recently, Stefan has begun a new position as a W2 professor for quantitative proteomics at the University Medical Center. Tenzer's lab strength in mass spectrometry and focus on the development and optimization and application of methods for quantitative proteome has been beneficial to a number of research areas. In a more recent method development project, the Tenzer lab has embarked on a mission to optimize the approach for correct determination of phosphorylation sites using latest mass spec and data analysis technology. From current research, this remains to be a challenge in the industry, especially for co-eluting and isobaric phosphopeptide isomers, which are phosphorylated on different residues. To provide more details about this work, we're thrilled to have Stefan discuss how single-shot phosphoproteomics and ion mobility-based mass spec can be harnessed to elucidate isomers using PASEF-DDA and PEAKS. Let's take a deep dive into how IMS-based proteomics can further our knowledge of cellular signaling and refine the view of phosphoproteome. I will pass my screen sharing to you now, Stefan, and you can begin when you are ready.
Okay, Zach, thank you very much for the very nice introduction. I'm thrilled to be here and to be able to present some of our latest research, which was done in collaboration with both the PEAKS team and also with the application specialists from Bruker in Bremen. And I'm going to present you today some of our latest results stemming from this very nice collaborative effort. The talk is going to be about how to achieve deep coverage phosphoproteomics without any fractionation upfront the LC system. And this has been enabled by using the PASEF-DDA technology from Bruker in combination with data analysis from PEAKS. So we initially planned to have the talk 1 week ago, but due to shutdown academia, shutdown STEM, which was supported by PEAKS, we postponed the talk to this week, and I'm very happy that we were able to support this, and I hope you have enjoyed the week and are now looking forward to the data we can present you today. The data I'm going to present you today is basically from acquired and analyzed in our group, and the people marked in green, this is Ute Distler. She manages our facility and the instruments, and Thomas, a PhD student who did a lot of the analysis in this project. So when we look at current challenges in phosphoproteomics, we know that most instruments, state-of-the-art phosphoproteomics workflows, are able to provide deep coverage and also mostly confident phosphosite localization. There are different enrichment strategies such as titanium dioxide, various IMAC technologies, various other fractionation, and there's of course a lot of different instrument platforms, TOF-based, like the timsTOF Pro or Orbitrap-based platforms. And obviously there's also a multitude of acquisition schemes to acquire optimal data sets, as for example, using chromatogram libraries as recently described by Brian Seal and Lindsay Pino. Basically, to do phosphoproteomics, we typically start for a bottom-up workflow with a protein mixture which is digested by typically using trypsin into tryptic peptides. These tryptic peptides are a mixture of non-phosphorylated peptides and phosphorylated peptides. And as phosphorylation is substoichiometric, meaning that not all phosphopeptides of a certain kind are phosphorylated, it is typically required that you enrich these phosphopeptides before doing an LC-MS analysis of these samples to achieve a reasonable coverage of the phosphoproteome. Just finding the needle in the haystack is easier when diminishing the size of the haystack and removing everything you're not interested in, meaning the non-phosphorylated peptides. I think that is well known. But there are, of course, current challenges that have partly been resolved and not completely resolved yet. So you can imagine there are peptides that have exactly the same sequence and also have exactly the same mass, but due to being phosphorylated by different kinases under different circumstances or for different challenges of the cells, you can see that there's, for this peptide having 4 potential phosphorylation sites on serine and tyrosine residues, there is a multitude of possible phosphorylation sites. This is a graphics from the recent publication with Rebecca Jensen, Nature Communications, nicely illustrating that all of these peptides, they have the same mass, they have the same number of phosphorylation groups, and they have partly different diagnostic fragment ion that can pinpoint towards one or the other of these peptides being present in the sample. This is, however, only possible if, when using a standard workflow, if these peptides are somehow separated, at least a little bit by LC separation. Because if they are completely co-eluting, you will obtain mixed chimeric fragment spectra, and having 2 of these positional isomers co-eluting, being co-fragmented as they have the same mass, you will end up with a mixture of diagnostic fragment ions that may or will likely make the deconvolution of these positional isomers completely impossible. However, we know since a few years that ion mobility plays an important role, as the separation in the gas phase can separate peptides not on their mass to charge ratio, but on their shape in the gas phase. And you might imagine that having phosphorylations at different sites in the peptide changes the conformation in the gas phase of these peptides, and thus makes them amenable to separation by ion mobility in the gas phase. And this is what we thought would nicely work. And obviously, when you have such an idea to test that, you also need nice samples to verify that. And here we were very lucky to be able to team up with people from our children's hospital who provided us with cells from a patient who had an 8-year-old child having osteosarcoma. This is a very bad prognosis for the patients. It's a very common cancer starting in the bones, and a lot of these cancers metastasize and are partly resistant to chemotherapy. And when they metastasize, the survival rates are pretty low, between 15 and 30%. And so so there's a high medical need to find out the signaling pathways leading to this malignancy and leading to dedifferentiation of these cells, and also determining how these different cancers respond to chemotherapies. So what we obtained is basically patient-derived cells from these osteosarcomas from a lung metastasis before and after a treatment with a multi-kinase inhibitory treatment scheme. We obtained these cells, sliced the cells, did an in-solution digestion followed by C18 purification to get clean samples for the phosphopeptide cleanup, which was in this case to show reproducibility of the workflow. We did enrichments in triplicate using titanium dioxide for both of the conditions. And these 3 enrichment replicates were then analyzed in technical replicates on a Bruker timsTOF Pro platform using PASEF-DDA. For the LC system, we used a nanoLC from Bruker using a 25 centimeter, 75 micron ID column, and quite a standard gradient going from 2 to 37% in 100 minutes at a flow rate 400 nanoliters with a column temperature of 50 degrees. For the PASEF acquisition, we used, as we wanted to do label-free quantification of the phosphopeptides in the end, we used a relatively short cycle time to accommodate for the high peak capacity of that column, and covered a precursor mass range from 100 to 700 m/z. And basically the entire ion mobility range covering any multiply charged peptides. We typically, in this acquisition scheme, use 10 PASEF scans with 100 milliseconds each, combined with a precursor scan to get the precursor ion information. And in each PASEF scan, you can acquire up to 12 MS-MS spectra, translating into about 120 or 110 MS-MS spectra per second. Notably, these MS spectra, even if acquired 100 Hertz basically, are still high resolution as the TOF is not affected by short cycle times. This is a contrast to an Orbitrap setting where the resolution goes down with shorter cycle times. For the data analysis, we processed these data in peaks using quite standard parameters suggested for PASEF-DDA data. So we used a 15 ppm precursor mass accuracy and a 0.3 Dalton mass accuracy for the fragment ions, trypsin with maximum 2 missed cleavages, and allowing phosphorylations and oxidations as variable modifications here. We processed this using rather stringent criteria, using 1% FDR on the peptide level, translating into about 220,000 peptide spectrum matches at a 0.4% FDR on the peptide spectrum match level. When we look at the results of these data, we see that in each single run of the samples, we were able to identify between 15,000 and 20,000 peptides. Of those were about 75 to 80% phosphopeptides. Overall, across the 6 samples we acquired, we were able to identify close to 28,000 phosphopeptides, taking basically everything that's identified. Obviously, when you look for positional isomers, you want to focus on peptides having a high A score above 6, which is 18,000 peptides, or even being more stringent with an A score above 20, which leaves us with about close to 12,000 peptides. When you look at detected positional isomers in these samples, we found about 6,000 pairs, which had both members having an A score above 6. And looking at these pairs, you see that about 2,000 of those are eluting within 1 minute, which makes them pretty much inseparable by classical LC. When we look at these positional isomer pairs, they obviously have exactly the same precursor mass to charge ratio as they have the same modification, but on different side residues. And when you look at how these basically differ, we have about 2,000 of these eluting within 30 seconds, which would not be basically separatable by classical LC with a typical DDA workflow on classical instruments. But on top of that, we obviously have ion mobility as an additional dimension of separation. And of these co-eluting phosphopeptide isomers, about 25% were perfectly baseline separatable by ion mobility separation in the gas phase. There are obviously other peptides that are also separated by mobility, but not co-eluting. These you would be able to be separated by LC alone, but these 457 phosphopeptide pairs were not separatable by classical DDA-based data sets. There is obviously a large number of positional isomers that are both co-eluting and also have very similar mobilities. To resolve that, we definitely need better performing instruments in the future, or alternatively apply pre-fractionation techniques offline before the LC separation to be able to separate these potentially offline. One of these positional isomers was identified from SRMM2, which is a matrix protein. It's a pre-mRNA splicing factor, and it has been shown already in previous studies to have aberrant expression in neurodegenerative disorders, and it's also implied in other carcinoma types, such as thyroid cancer or liver cancer. Previous studies have already shown that aberrant phosphorylation plays a role in tumor pathogenesis, and we were able to get a very nice coverage on phosphoproteome level of these proteins, and also were able to identify several co-eluting phosphopeptide isomers. Here's an example from our data. Basically, we look in the second half of the protein at position around 1,700 amino acids, and there is a peptide that carries 2 that carries two phosphorylation sites, phosphorylations on different residues, and those are basically co-eluting. Two of them are completely co-eluting, and one is eluting about half a minute later. When you look at them, these are basically harboring phosphorylations at the different positions in the protein, and they might have different functions, and some of them were also differentially regulated between the chemotherapy treated and the normal phosphopeptide samples. Focusing on the peptides having an A score above 20, we had these three peptides outlined here, which are high confidence information, and we were able to show by using PASEF-DDA that they are really nicely separated in the gas phase. Here you look at an RT-M/Z plot, which shows basically the different scans applied here, and you can see that these two spectra are giving you differential fragmentation spectra that can allow us to assign phosphorylation sites to specific residues on these peptides. This is only possible, keep in mind, as they are co-eluting, as the ion mobility separation in the gas phase allows us to pull them apart here. These are the two sequences we obtained from the PEAKS search. You see these two peptides share a common phosphorylation site on this serine residue and have the different phosphorylation at either serine number 2 in the peptide or serine number 3 in the peptide, corresponding to position 1690 or 1691 in the peptide. Wasn't it quite nice and quite surprising for us that these two peptides that just shift the position of the phosphorylation by just a single amino acids result in a baseline separation in the ion mobility space. So yeah, actually the ion mobility provides us here with a unique opportunity to unravel site-specific phosphorylation in tumor-specific, tumor-relevant proteins at a very high resolution. And this would not have been possible using any other instrument at the moment. So obviously, you see that these are separated in the gas phase on the mobility. And when we look at the MS1 spectra, you see they have exactly the same precursor mass. They have exactly the same retention time, but their mobility differs quite significantly, 0.93 versus 0.96. And these were picked up when we look at the mobility versus M/Z plot. They were picked up at different positions in this plot. So you see they have exactly the same precursor charge here, but they are picked up at different mobilities. They have the same retention time, keep that in mind, and they produce different fragmentation spectra. This is not only one cherry-picked example, but we have a number of other things. One very nice one was from the A kinase INKA protein, showing a phosphorylation in this residue, which was significantly changing after treatment between the two samples. So again, here we see the two different fragment spectra indicating phosphorylation on two different residues, which are just adjacent residues again. And again, in this case, we see that they are co-eluting in retention time quite perfectly, so they are indistinguishable on retention time, but in this time they are not completely baseline separated, but still nicely separated in the gas phase. Again, in the normal MS without ion mobility separation, you would not be able to separate them perfectly. You would get mixed spectra, as obviously they are co-fragmenting and give you chimeric spectra, and would not allow you to basically resolve these two different phosphorylation sites. Sorry, excuse me. To analyze the entire data set, we made use of INKA analysis, which was done in collaboration with our with the Netherlands. And here we basically feed quantitative proteomic data and phosphosite data into the workflow and are able to identify different proteins significantly changing after drug treatment. To sum up what we've been doing, we can show that the PASEF-DDA phosphoproteomics are able to provide us with a deep phosphoproteome coverage. And keep in mind, we identified about 28,000 phosphopeptides without any offline SCX reversed phase, whatever fractionation. So this allows us to give you a complete picture. And this is only possible because we use ion mobility to detect these co-eluting positional isomer pairs. These data allowed us to define altered kinase activities before after the treatment with kinases, and we obviously integrate the information of proteomics and phosphoproteomics, and will in the future also complement these data by specific enrichment of tyrosine phosphorylated peptides using specific antibodies. Last but not least, I want to thank all the people involved here. From my lab, as told before, Ute Dissler and Thomas Michner did most of the work. Ruben is our technician who is basically taking care of all our sample prep here. We had a very nice collaboration making this data possible in collaboration with Bruker, Stefanie Kasper-Schönefeld, Christina Marx, and Scarlett Koch. I want to thank the people from the Children's Hospital in Mainz, especially Claudia Perret, who provided us with the samples and discussed a lot of the data. And obviously to Conny Jimenez and Alexander Hennemann, who helped us with the INKA analysis and put in a tremendous amount of work to make that analysis work for our type of data. Funding was from DFG, SFBs, and various local funding options. And with that, I want to thank you for your attention, and I'm happy to take questions.